Okay, well, I'd like to start by thanking Robert for the very kind invitation to come and speak to you today. So as you've heard, I'm from the University of Bradford in the UK. A lot of you probably haven't heard of Bradford or don't know where it is. So Bradford's roughly halfway between London and Scotland, so pretty much in the middle of England. And it's pretty much at the spot where Emily Bronte wrote Wuthering Heights 170 years ago, and you can see that the university certainly looks the part. So my background, as you've just heard, is principally in molecular biology. So I'm not a clinician. I'm a sort of basically my background is as a bench scientist. So this talk contains a lot of molecular biology, for which I apologise in advance. But I'm going to try to approach it in a way that makes at least makes some sort of sense out of it. One of the things that we were very interested in about 10 years ago were a group of genes called the HOX genes, and that accounts for this rather strange picture we have at the beginning here. In fact, this in the field of molecular biology and of genetics and of developmental biology, this picture is actually becoming something of an icon because it represents some of the really brilliant science that went towards understanding how genes determine who we are, how they pattern the early embryo. And this is one of the pictures which won Marianne Nusslein-Volheid and her colleagues Nobel Prize for medicine. And it shows the result of what happens when one of these genes goes wrong. So this is the head of a fruit fly, and you can probably see something's fairly strange about it. And what's strange is that instead of having antennae on its head, it has a pair of legs, and this is the result of a single gene called antennapedia, one of these so-called HOX genes, being expressed in the wrong place, and is expressed in the cells that will eventually make up the eye instead of another gene, and the result is a leg. Now, one of the other things you kind of see from this picture is that the legs are actually fully formed. They're perfectly normal legs, and in fact, if you're a fly, you'd probably be quite happy with these legs, except of course that they're stuck on the front of your face. But apart from that, things aren't looking too bad. So the reason why I have this slightly tangential introduction about HOX genes through the fly head is because it makes a very important point. Well, it makes two very important points. One of these is that HOX genes are involved in determining the identity of cells and of tissues, not just of flies, but of vertebrates as well, including us. But the other, which is far more relevant, it turns out, to cancer, that in addition to determining the identity of cells, they also help to drive a program of rapid cell division, because obviously if you're going to go from a very small number of cells in the early embryo to form a leg, then one of these things the cells have to do is undergo numerous rounds of proliferation. They have to divide a very large number of times to form a leg. And in addition, they have to survive. A lot of cells in the early embryo actually die as part of a natural process of development, and the cells that have formed this leg likewise have managed to avoid cell death. So antennapedia and the other HOX genes do these two very important things, which are obviously relevant to cancer, which is to drive cell division and to ensure cell survival. So there's just one more introductory slide about the HOX genes. Just explains how they've changed over the course of evolution, but essentially we have the same genes as flies from the point of view of HOX genes. Flies have 8 HOX genes, which are expressed in a pattern along the anterior to posterior axis, where they determine which part of the fly is which. And likewise in mammals, including us, there are now 39 HOX genes, which are very closely related to the fly genes, and likewise these are also expressed in a pattern from the head down to the tail, where they determine cell identity. So this is all very interesting, but one of the questions that we asked, along with others, about 10 years ago now, is whether the HOX genes are also involved in cancer. And clearly they're a likely candidate as they promote cell division and cell survival. But if they are, they're also potential targets and markers in cancer, because we know that the vast majority of HOX genes are actually silenced before birth. So they're important during development, but they're not expressed in most normal adult cells. So the question was, are they turned back on in cancer, in tumours? And to cut a long story short, they are. And this timeline, which is just included on one of the slides for you, which I'm not going to dwell too much on, but it essentially goes through our understanding of the role of HOX genes in cancer. And it's based on a review published 6 years ago now in Nature Reviews Cancer by Sukumar and Shan. And the place that we come in is here towards the end of the story, which is really where part of our story picks off, starts off in 2007. But you can see that this has actually been a long process of recognition of the role of HOX genes in cancer, starting with their role in leukemia, where they are both switched back on in leukemic cells and expressed at very high levels, but they also form chimeric partners with other genes to drive oncogenesis. And then subsequently it became clear that HOX genes are expressed also in solid malignancies. And this includes mesothelioma, and in fact mesothelioma is one of the malignancies where you see the greatest degree of HOX gene dysregulation. In other words, mesothelioma is an example of a cancer where you see a large number of these HOX genes being switched back on compared to normal cells of the mesothelium. And this figure just essentially shows an example of this. So these are different these are different mesothelioma tumours taken from patients. And it's an extremely compact figure, and the details don't really matter that much. But it's looking at a group of these HOX genes which are known to have the most significant roles in terms of promoting cancer, in terms of having oncogene-like properties. And what you see is that a lot of these tumours do show significant overexpression of the HOX genes at one level or another. It varies a lot between each tumour, and there might be very good reasons for that, which I'm not going to talk about today because there's not enough time. So almost like a fingerprint on each tumour, this level of expression. And yet you do see the majority of tumours show this very high level of dysregulation. So this is both at the RNA level but also at the protein level. So here we have some immunohistochemistry, and it shows that the proteins from these different genes are correspondingly also highly expressed in these mesothelioma tumours. So when you dig deeper, you actually find clues that these HOX genes, or at least some of these HOX genes, do indeed have an important role in the biology of mesothelioma. And this was part of the study which looked at the significance of the expression of different HOX genes in mesothelioma. So this was a large set of patient tumours looking at the influence of HOXB4 on patient survival, and indeed other HOX genes. So this is one of the 39 HOX genes, HOXB4. But what we see is that that patients who have tumours that express very high levels of HOXB4 have significantly worse survival than patients whose tumours only show low levels of HOXB4. So this also points to a role of HOX genes in the biology of mesothelioma. And again, this is one of the more complex sort of molecular biology type slides, so I'm going to skip over it. And all I'm going to say about this is that there are a number of HOX genes which now are known to have oncogenic functions. In other words, they support the growth and survival of cancer cells. And in this study, we're primarily looking at 5 different cell lines derived from mesothelioma, and we can see that all of them express these oncogenic HOX genes to different levels. There's also evidence from the literature that a number of HOX genes actually do the opposite. They have tumor suppressor functions. In other words, they actually block the growth of tumours. And we can see that some of these cell lines, indeed some of the primary patient tumours, also show very high levels of these tumor suppressors. But we'll think a bit more about that in a minute. Okay, so this is all very interesting, but from an experimental point of view, and also ultimately from a clinical point of view, it leaves us with quite a difficult problem, and that is that there are 39 different HOX genes, and as we saw in those previous figures, that many tumours overexpress multiple HOX genes, and some of them show the overexpression of 30 or more different HOX genes. And so obviously conventional molecular biology approaches, and I know that a lot of you will have to take my word for this, but conventional molecular biology approaches really only allow you effectively to look at the function of 1 or 2 genes at once using techniques designed to knock down gene expression. So it leaves the problem of how can we establish whether or not all of these HOX genes together have some combined effect in promoting the growth and survival of cancer cells. As we know from embryology, for example, that a lot of these HOX proteins actually have redundant functions. In other words, a lot of them do the same thing. So if we just knock down one of these HOX genes, how are we going to find out what they actually do? So the approach we took, work that started, as I say, about 10 years ago now, was to try to block the interaction between HOX proteins and one of their cofactors called PBX. So HOX and PBX are 2 different proteins, and they need to bind together, they need to join together in order to bind to DNA and to change gene transcription, which is essentially the business end of what they do. They're these so-called transcription factors that modulate the way in which genes are turned on and turned off. So this picture here shows a computer simulation of HOXA9 or HOXB1 binding to PBX. And what you notice is that although both of these proteins, as you'd expect, have very complex structures, the actual interaction between them is mediated by a very short segment. It's mediated by only 6 amino acids, a so-called hexapeptide. So this is a very short part of the protein on HOX, which binds to a pocket in PBX and actually mediates this interaction between HOX and PBX. So the question was, is there a way that we can disrupt this interaction? And if we do that, can we then work out whether HOX proteins have a function in mesothelioma? So to do this, we developed a HOX PBX antagonist. So this initially was a peptide which disrupts the binding between HOX and PBX. And it is simply the hexapeptide sequence of these 6 amino acids on HOX, which bind to the pocket in PBX, together with a polyarginine sequence, and this part of the peptide simply allows it to be taken up by cells. And for a lot of these slides, we talk about a control, which is called CXR9. So this is a negative control, and what CXR9 is exactly the same as HXR9. It simply has a single amino acid difference. This tryptophan is changed to an alanine. And to cut another fairly long story very short, the answer is that yes, HXR9 can indeed disrupt the binding between HOX and PBX proteins. So this little schematic just shows you how. So HXR9 is this small peptide. It's taken up by cancer cells. It binds to PBX and blocks this interaction between HOX and PBX. And as a result, target genes, HOX PBX target genes, can no longer be regulated by these HOX-PBX combinations, these so-called HOX-PBX dimers. And this is just some of the more complex molecular biology that I'm going to go over very quickly, which proves that it works. This is a technique called immunoprecipitation, and it's designed to look at how proteins interact within cells. So what we've done is we've found a way of looking at this complex between a particular HOX protein, in this case HOXD9, and PBX, by using beads, so we can extract this pair of proteins from cancer cells. And what you can show is that when you add HXR9, you can no longer recover HOXD9, although you can still recover glucocorticoid receptor, which also binds to PBX. Whereas if you add CXR9, this control peptide, which is a single amino acid difference, you see that you get HOXD9 back. So that single amino acid is in fact crucial to the function of HXR9. And this is the results of another published study that essentially show the same thing. But I'm just going to show you quickly the results on the bottom, because I think they're particularly striking demonstration that this actually works. So these are some cancer cells here, and the blue color is the nucleus of the cancer cell. PBX is shown in green fluorescence, and HOXA9 is shown in red fluorescence. And what you find in that cells that's treated with this control peptide, that the HOXA9 and PBX bind together to form this dimer, so you get the combined resulting signal throughout the cell in the nucleus and in the cytoplasm. But it's quite striking that in the cells that are treated with the HXR9 peptide, you only get the HOXA9 signal. That HOXA9 is completely excluded from the nucleus of these cells because it can no longer bind to PBX. So that's what HXR9 does then. It prevents HOX and PBX proteins from binding together. And again, this is another little bit of molecular biology which seems in some ways needlessly complex, but I've included it because I needed to think again about this idea that some HOX proteins are acting as oncogenes, so they promote tumor growth, and others act as tumor suppressors. And just very briefly, the point that we're making here is that in fact the HOX-PBX dimer generally is required for the activation or the suppression of oncogenes, whereas the single HOX proteins and not the PBX dimer is required to activate genes such as P53, which is another so-called tumor suppressor, a very important tumor suppressor, which blocks tumor growth. So I've lost all track of time because the timer stopped. So if you just let me know when I've got 5 minutes, that would be that'd be great. Okay, right. So HXR9 disrupts HOX-PBX dimers, and as we'll see in a minute, kills mesothelioma cells, which is obviously why I'm here today, and also blocks the growth of tumours. But one of the very important things that's become apparent over the last 5 years in medicine is the idea of personalised treatment, of personalised medicine, whereby we can select which patients are going to benefit from a particular type of treatment. And in order to know which patients are going to benefit from a particular drug, we need things called biomarkers, which are essentially, which are frequently genes, specific genes that are either expressed by the tumour or that change in expression when they're exposed to a certain drug. And I'm just going to give a few examples in this talk where at the very early stage in the lab we've tried to develop these biomarkers which will allow us to do a number of things. They'll allow us to monitor the response or potential response to treatment by HOX-PBX inhibition, but also allow us to select which patients are most likely to respond to that treatment. again, this cuts quite a long story short, but what it shows is that you add HXR9 to these cells, that a number of genes change very rapidly in expression, and one of these is called CFOS. And in fact, it turns out that this very rapid expression in CFOS, and it is very rapid, it occurs within about 60 minutes of adding HXR9. So compared, for example, to drugs like cisplatin, where you don't really see any effects for at least 3 days, it works very quickly. And this very rapid increase in CFOS is actually one of the mechanisms by which HXR9 kills cancer cells. So some of these cell lines, but not all of them, but some of them are, some of these mesothelioma cell lines are sensitive to killing by HXR9, as you've probably already gathered, and this slide just summarizes that data. And this is primarily through a process called apoptosis. So this is a programmed cell death that can be induced in cancer. And again, this is more sort of technical molecular biology type thing, but the importance of killing cancer cells, or potential importance of killing cancer cells through apoptosis, is that it reduces the degree of inflammation that might occur from killing cancer cells. Now, sometimes inflammation when you kill cancer cells is good, of course, as we've already heard, and we're going to hear some more about. I think that inflammation can induce an immune response, so that can be good. But there are also lots of times when you don't want inflammation when you kill cancer cells, and certainly some of the applications outside of mesothelioma where we're thinking about disrupting HOX PBX require that. So primarily this cell death is driven by the induction of apoptosis. So this is looking at some mouse models now. So this is asking the question, if we block HOX PBX using this peptide, HXR9, can we block the growth of mesothelioma tumors? So this is an experiment in mice which have mesothelioma tumors. And what we've done is that we've let them grow up to a size of about half a cubic centimetre, and then we've given these mice a single dose of 25 mg per kilogram HXR9 into the peritoneum. So this is IP. And what you see at this point is that you see a very significant reduction in tumour growth. And another interesting finding actually from this mouse model, which corresponds very nicely to what we saw with the patient data with HOXB4 being associated with shorter survival, is that the actual size of the control tumours, these are the untreated tumours, there's a significant relationship between the level at which these tumours express HOXB4 and the final tumour size. So again, it suggests that HOXB4 and the other HOX proteins are involved in driving tumour growth in mesothelioma. And this coming back again then to this idea of personalised medicine. So if disrupting HOX PBX is a useful method for treating mesothelioma, then is that going to be true for all mesothelioma patients? And the answer is probably not. And if not, then how can we predict which patients are going to best respond best to HXR9? And this was a study looking at potential biomarkers again, potential ways in which we can tell which tumours after biopsy are likely to respond best to HXR9. And what we've done is we've looked at a subset of these HOX genes in the tumours, and we've asked, is there a relationship between the expression of these HOX genes and the sensitivity of tumour cells to HXR9? And again, cutting quite a long story short, the answer is yes, there's a very strong relationship between sensitivity to HXR9 and the expression of this subset of HOX genes. So it implies that there's a way of predicting which patients will best respond to the treatment. And then in addition, some of the other work we've done implies that there's a useful marker of response. So often in clinical trials, you want, if possible, an earlier endpoint, as the so-called surrogate endpoints clinical trials, which tells you whether or not the tumour has actually responded to the drug that you've used. And this was just a study looking at some of these mouse tumours with a gene called DUSP1, which is another gene that responds to the addition of HXR9. And you can see that when you treat these tumours with HXR9 or a variant of HXR9, which is more effective, it turns out, called HDL0001, you see a very strong induction of DUSP1 a few days after the mouse has been treated. So this looks like a potential surrogate marker that could potentially be used to find out early on whether there's been a response to treatment. Okay, good. So how long have I got? Is it negative? Huh? Is it negative? Okay. Anyway, right. So that's the end of the first part of the talk. I'm just going to very quickly talk about development of small molecule inhibitors of HOX PBX. To finish off the peptide story, HXR9, or in fact a very similar peptide to HXR9, is hopefully going to go into phase 1 trials next year. So we're almost there with that. But generally, small molecules are preferred to peptides. So the other question we've asked is, can you develop a small molecule inhibitor of HOX PBX? And generally, this is quite difficult with transcription factors because they tend to interact through very large planes, very large hydrophobic surfaces. But there's an indication here that it might be possible, because you remember the difference between HXR9 and the control peptide, which is completely inactive, is only a single amino acid. And in fact, this corresponds to what you see in the molecular modelling. It turns out that single amino acid, the tryptophan, seems to be one of the most essential contacts between HOX and PBX. And again, in this model, what we've done is to overlap lots of different HOX proteins with PBX. And in every single case, you see that this tryptophan, this conserved tryptophan, is pushed into the same position relative to PBX. So it's indicating that there's actually a very specific molecular interaction between these two proteins. And what you can do then in the conventional ways to develop pharmacophores which allow you to predict which drugs, which small molecules will potentially act as inhibitors of this. And in turn, this allowed us to develop a library for testing consisting of about 1,000 compounds in the first instance, because as with all computer modeling, especially this computer modeling, the results aren't really that specific, so you end up with about 1,000 potential hits. And this is just a screening assay, a fairly complex screening assay, but it's a fairly complex interaction, which allows you to screen for which drugs will block the interaction between HOX and PBX. So this allowed us to screen through this library, which in turn told us a number of things, but identified a number of drugs which do block this interaction between HOX and PBX. And in fact, they do it quite specifically, and they also induce this biomarker that we talked about earlier, CFOS. So again, this project is lagging somewhat behind the peptide project, which is really in many ways proof of concept, although it has actually made it through now to hopefully to clinical trials next year. But we're making some good progress with this as well. Great. You've been very patient. Thank you for your attention. This is just to prove that the sun does sometimes shine in Bradford, although it's relatively rare. So thank you.